In this video, we will discuss the process of phagocytosis. We will discuss how a bacterial cell gets attached to the phagocyte. After that, it enters inside the phagocyte and forms the phagosome. We'll discuss the role of lysosome in degrading the pathogenic microorganism. We'll discuss the process of phagocytosis in detail, and I hope that this video is going to help you to understand this process very clearly. Before we move on to the lecture part, I have a quick request to make. If you are new to the channel, then please do subscribe to the channel and show your support. If you like the video, hit the like button. Today we're going to understand the process of phagocytosis. This is an important process of our immune system. To explain this process, first I need to design a phagocyte. So what you're seeing right now is me designing the outline of the phagocyte. Now the outline is complete. Next thing I'm going to design is the nucleus, which is the most important part of the cell. After that, there are some additional organelles like mitochondria. I'm also going to design some other important organelles like vesicles, endoplasmic reticulum. So endoplasmic reticulum is very important because it plays an important role in case of protein synthesis. So to explain this process, First, I'm designing the phagocyte, and then I'm going to discuss this whole process in 5 distinct stages, where I'm going to show you how bacteria attach, and then it gets processed, and then the final product is released. Right now you can see in red, I'm designing the bacterial cell. This is considered as a pathogen, specifically bacterial cell. So what's going to happen, this bacterial cell is going to be sensed by the phagocyte using various receptors that we already discussed in our previous videos. After that, the cell is going to construct pseudopodia. So this is the step 1, which is bacterium attachment to the membrane or to the cell. So let me write it down: bacterium attached to the membrane using pseudopodia. So these membrane extension you are seeing that I have designed close to the bacterial cell, these are pseudopodia. This is the step 1. And then what is going to happen, that this bacterium is going to be ingested by the cell, and it will be contained inside a membrane-bound structure known as phagosome. So this will be the step 2, where bacterium will be ingested. So let me write it down like bacterium ingested inside the phagosome. So this is the step 2, where you have bacteria contained inside a membrane-bound structure which is known as phagosome. Now let's move on to the step 3. In the step 3, you will have lysosomal organelles that are present inside the cell and plays very important role in case of the digestion of the bacterial cell. So in stage 3, what will happen is the phagosome will merge with the lysosome, constructing phagolysosome. So let me write it down: phagosome plus lysosome constructing phagolysosome. And inside phagosome, lysosomal enzymes are going to digest the bacterial cell. And in the fourth stage, what is going to happen? These enzymes, they're going to lyse the bacterial cell. So let me write it down: digested by lysosomal enzymes. So inside the phagosome, the bacterium will be lysed. And in stage 5, the byproduct will be released outside the cell. Basically, the bacteria will be killed and the products will be released outside the cell after processing. So this is the complete process of phagocytosis, where the pathogenic organism, in this case bacterial cell, will be processed in multi-steps. Step 1, where bacterium will be attached to the phagocyte. Step 2, where the bacterium will be ingested. Step 3, where phagosome will be merged with the lysosome. Step 4, the lysosomal enzymes, they will digest the bacterium. Step 5, the final product, the lysed bacterium, will be released from the cell. I hope now the process of phagocytosis is clear to you. I hope this topic is clear to you. We have discussed how the bacterial cell gets killed by the phagocyte. We have discussed how the process of attachment and then the processing, further processing using lysosome, takes place inside the phagocyte. Because of the phagocytosis, our immune system keeps the pathogenic organisms out from our body. I hope this topic is clear to you, and if you like the video, then please hit the like button, and if you're new to the channel, then please do subscribe to the channel.